Good morning, everybody, and welcome to this first seminar of online seminar of open science of the Italian Reproducibility Network. We are very pleased to do the kickstart of these year's seminars, and we are very happy to have you all here. Let me first thank you, the Italian Association of Psychologists of Psychology, who support our activities, and we very thank you them for this support. And then let me ask to Davide Crepaldi, who is the founder and president of the Italian Reproducibility Network, to say some words on today's seminar. So thank you, Davide.
Thanks a lot, Michela. Thanks, everyone, and welcome also my side. This wonderful seminar series, and particularly the opener of the seminar series. So even more important that you're all here and that the community is gathering around these events, which I think are very, very important to promote a better research culture in our community. And to this end, together today, we're very, very happy to have Marcus Munafò with us. So it's very, very honored to have you here, Marcus. Thanks a lot for accepting our invitation. So Marcus is a professor of biological psychology in the School of Psychological Science and the MRC investigator in the MRC Integrative Epidemiology Unit in Bristol. He co-directs a large research lab together with Angela Atwood, Olivia Maynard, and Ian Pentonvoke. And of course, is a very long-standing interest in factors that contribute to research quality. From this interest of his, he's gained a very prominent position in the movement that is trying to improve the way that we do our research. And recently has also been appointed Pro-Vice Chancellor for Research Culture at the University of Bristol. More widely than his home university, of course, Marcus is probably best known for being the founder of, co-founder, maybe I should say, of the UK Reproducibility Network, which is a little bit like the father, or at least the older brother of all the reproducibility networks that have spread since then. The ITRN was launched in 2019. And now we are some tens of reproducibility networks around the world, in Europe, and actually more widely too. And of course, the Italian Reproducibility Network, we have a little bit of debt to Marcus for many reasons. And there's also a nice sort of anecdotal connection. Marcus was invited in 2020 to give a talk by Vittorio Iacovella and Marco Minusi at CIMEC. Well, actually it was online because we were in the full swing of the pandemic, unfortunately. And this is one of the moments where this idea of founding an Italian reproducibility network came about. So we're also particularly thankful for this, Marcus. Okay, the stage is yours, and thanks again for accepting the invitation.
Well, thank you very much for inviting me. That was a very kind introduction. So I'm going to talk today about where we've come from in terms of the problems that motivated me and others to start thinking about ways in which we could improve the quality of the work that we do, and what we are doing now to actually bring to life some of those proposals for how we can work differently, and also to what extent we can learn from other industries, other sectors, as well as share what we do well within academia with those other sectors. And I'm going to start with something that is a little bit autobiographical. Before I do, these are my disclosures. None of them are really directly relevant to what I'll be talking about now, except that as chair of the UK Reproducibility Network, I clearly have a particular personal perspective on whether or not there are issues to address and how we might go about addressing those issues. But I should also say that the project I'll talk about towards the end, which is about developing a quality framework in academia and in academic research, particularly in the life sciences, comes from this philanthropic donation that is mentioned at the end of that disclosure. I don't think I need to say more on any of that. Now, I'm going to talk principally about scientific research. I think much of what I talk about is relevant or can be relevant to other scholarly disciplines. And one thing that certainly the UK Reproducibility Network is trying to do, and other reproducibility networks as well, is to think about how we can be relevant across a broad range of disciplines, and how, for example, open research can be implemented in very different domains, ranging from particle physics through to art history, for example. But for the purposes of today's talk, I'm going to focus on the life and biomedical sciences, psychology being one of those. And I do that in part because this is really where the conversation started. At least in the UK, the motivation behind establishing the UK Reproducibility Network came from concerns raised in psychology and other disciplines about the quality of work. And the concern is captured here, which is that we are taught as undergraduates that science is self-correcting, that the scientific method has a self-correction function built into it, and that whilst we will make mistakes, we will go down blind alleys, because when we are trying to advance knowledge, we necessarily will come up with findings that are spurious. Eventually, those findings will self-correct. So we don't need to worry. And this, I think, is a challenge to us, because the question then needs to be, well, is that true? To what extent is science self-correcting? To what extent does it self-correct rapidly enough? And to what extent are we doing the things that allow science to self-correct? What are the things that allows science to self-correct? Running replication studies, publishing null results or results that go against what we expected to see, admitting when we're wrong. All of those things, I would argue, we don't do well enough. And therefore, science is not going to self-correct as rapidly as it might. And therefore, the speed with which we advance knowledge is going to be slower, because we'll be investing effort in these blind alleys that don't lead anywhere. And I want to start with an example of that is semi-autobiographical, if you like. When I was a postdoc, I was doing work on genetic influences on smoking cessation response. So why do some people respond better to nicotine patches or gum or whatever than other people? And in those days, you had to genotype manually. So you had to take a blood sample and go into the laboratory and pipette and so on. And it was expensive and it was time consuming. And so we identified specific genetic variants known as candidate genes that we wanted to explore. And so I became familiar with the candidate gene literature, particularly in the context of behavioral psychological phenotypes, and started to review that literature quite closely. And in the context of anxiety and depression, which was a sort of adjacent field that I was interested in, there was this incredibly rich and exciting story unfolding about the role of the serotonin transporter gene. And some of you may be familiar with this. There was an article published in 1996 suggesting an association between variation in that serotonin transporter gene and anxiety-related traits, suggesting a biological basis to individual differences in anxiety. And we've known for a long time that anxiety has a partial genetic basis. Then there were mechanistic studies looking at the extent to which that same genetic variant was associated with activation in the human amygdala, the region of the brain that responds to threat, and further studies looking at the extent to which that genetic variant might explain why some people react more strongly to stressful life events in early childhood than others do. So it was a really rich, compelling story. This made it into the textbooks. People were learning about this as undergraduates because it fitted together so nicely. And everything built on that original foundational finding of this association between the serotonin transporter gene and anxiety. There was only one problem, which is that it was all wrong. It was all wrong. And we know that with absolute certainty now, and I'll explain why in a moment. These are not my words. I put them into quotation marks because they come from this blog post, which is a review published a few years ago of the serotonin transporter gene literature. And there's this great quote within it from Scott Alexander. Scott Alexander is a psychiatrist. He writes this blog post on all sorts of different topics. And there was a review article that came out that captured his attention, and he wrote this blog post. And he was angry. He was angry that so much time and effort had been wasted on something that was clearly wrong. And it was fascinating to him that this rich story built up with all of these studies seeming to build on that original finding, when that original finding was a false positive. This isn't just an explorer coming back from the Orient and claiming that there are unicorns there. It's the explorer describing the life cycle of unicorns, what unicorns eat, all the different species, subspecies of unicorn, which cuts of unicorn meat are the tastiest, and a blow-by-blow account of a wrestling match between unicorns and Bigfoot. How did this false positive finding lead to such a rich and complex story with so many careers built on the back of those publications, so many grants funded to explore this avenue further, when there was nothing there. How do we know that there was nothing there? Well, the original study, the one that showed an association between the serotonin transporter gene and anxiety, was in 500 people, which by the standards of the time was quite large. But shortly after, so in 2005, and here's another disclosure, I'm an author on these papers, but there were other people publishing similar findings. In 2005, so less than 10 years after that original finding, there was a study in 100,000 people that was identical to that first study, just looking for an association between the serotonin transporter gene and anxiety-related traits. And there was no evidence of association at all in 100,000 people. So given that one is an identical replication of the other, I would suggest that the sample size of 100,000 is going to be the more robust finding than the sample size of 500. And there were also concerns around the gene-environment interaction literature, this idea that the genetic variant influences who responds more strongly to stressful life events. Again, in our review article, given reasonable assumptions, our simulations indicate that published studies are underpowered. This leads us to suggest that the positive results for the interactions are compatible with chance findings. So very early on, there were reasons to believe that this was a false finding, a chance finding. No evidence, no suggestion of malicious behavior, malintent. This was simply a spurious finding, and these are going to happen. But did the literature self-correct at this point? Did people say, okay, this looked exciting, this looked interesting, but it's very clear now that that basic association doesn't hold, therefore everything else falls down because you pulled away the bottom of the house of cards? This was, if you like, the self-correction event. And there were other things happening at the time that meant that credible behavioral geneticists stopped investigating this genetic variant. In fact, more generally, stopped investigating candidate genes. The move to whole genome methods, genome-wide association studies, meant that it was clear that the candidate gene approach just hadn't worked, and the genome-wide approach worked much better in terms of robustly identifying genetic variants. So we had an opportunity for science to self-correct, did we? I think you can probably guess the answer. This is a plot of studies in PubMed that investigate the serotonin transporter gene. You can see the first published study in 1996. You can see the self-correction event indicated in 2005 and a later one in 2008. And these are not the only studies, again, showing that that original finding was spurious. The majority of studies in this topic, in this literature, were published after we knew that that initial finding was a false positive. Why? Because scientists are incentivized to publish. We get rewarded for publishing. We don't get rewarded for being right. We get rewarded for churning out papers, making our CVs heavier, and publishing in certain kinds of journals in particular. And the fact that you could still publish these findings if you wanted to somewhere meant that people kept doing so. So eventually, science self-corrected. If you look at 2023, there are far fewer papers being published on this topic now than there were 10 or 15 years ago, but they are still being published. And an enormous amount of effort has gone into all of that work. In fact, we have a paper coming out at the moment, or that we're writing at the moment, on the environmental impact of all of that wasted effort in terms of the -80 freezers that you need to store the samples, the laboratory consumables, the time spent flying to conferences to give talks and so on. All of that generated by something that is clearly false and only serving to sustain scientists' careers, but not meaningfully advancing knowledge. So is science self-correcting? Now, how does this happen? And I talked about this before. Some of you will have heard me touch on this point. I think we need to remember that scientists are human, and humans are fallible. There's a nice book by Nicholas Taleb that talks about how we are not very good at understanding randomness. It's why people find p-values difficult to understand. Scientists may be in the business of laughing at their predecessors, but owing to an array of human mental dispositions, few realize that someone will laugh at their beliefs in the disappointingly near future. In other words, we see the world through a lens, and that lens is a personal lens to us that is influenced by our beliefs, our prejudices, our biases. And in the context of academic research, we go into science wanting to find something. That's a good reason to go into science, but it means we can be led astray because we can be fooled by noise in our data, fooled into seeing something that's not there because we want to find something. And at the same time, we work within incentive structures that shape our behavior in ways that are unhelpful. This is a nice quote from a survey that's quite old now. Certain features of the working environment of science may have unexpected and potentially detrimental effects on the ethical dimensions of scientists' work. Not ethical in the sense of people consciously trying to defraud the system or fake their data. That happens, but I hope it's rare. This is more the well-intentioned, well-motivated researcher who wants to find something, but also finds himself working in a system where if they don't publish, if they don't get grant, it's going to be difficult to get the next job. And my experience is that certainly early career researchers feel that tension very acutely. They went into science quite idealistic, and they become much more pragmatic when they realize that in order to succeed, they need to do certain things, like publish whether or not the results are particularly robust. And I think a third problem is that we work within still a very ancient culture. The culture of academia can be traced back hundreds of years to the Enlightenment, and the model of academia that we maintain within most universities in Europe, at least at the moment, is the 19th century ideal of the independent scholar who is drawn from a certain section of society, who is independently wealthy, who is pursuing knowledge for the sake of knowledge. That's the story that we tell about ourselves, even though the reality of academic life is very different now. So what does that look like? What does that mean? I think this paper, which again you may be familiar with, can help to illustrate that. So in this study, if you're not familiar with it, real data were collected on real people, undergraduate students, and they were randomized to listen to When I'm 64 by the Beatles or another type of music. And those that listened to When I'm 64 became a year younger. Not that they felt younger, but you turn back the arrow of time, you lost a year of life. And when you get to my age, that's a really exciting finding. But the reality is, of course, it's false. We can't turn back the arrow of time by listening to music. If there are any physicists in the audience, I think they would raise an eyebrow at that. The point is, this is structurally identical to any number of different human laboratory studies that are published every day. You randomize, you measure something, you infer causality from a difference between the two arms of the study. The study showed two things. One is that if you build enough flexibility into the design, conduct, and analysis of your study, you can get a small p-value very easily just by running lots of analyses and eventually choosing the one that gives you that small p-value. And that much is worth knowing. But I think the more important point that this paper makes is that then as an author, you have a choice. You can choose to write up your study fully transparently, describing everything that you did. And this is the full abstract shown there. So describing all of that flexibility in the design, conduct, and analysis of your study. And if you read all of that flexibility, you come to the end and you go, well, of course they got a small p-value because they ran 100 tests. And that one small p-value doesn't mean anything in that context. But you have another option as an author. You can write up a compact, redacted version of what you did, where you don't describe everything that you did, you just describe enough to get to that final p-value. And that's shown in bold, that much shorter abstract that is not intended to give a full account of everything that you did. It's intended to tell a story, convince the reader, convince the journal editor that this is an exciting finding. So you have that choice. You can give a full account that is full and transparent, or you can give a shorter account that misses out some key information. And the question for us is, as a reader, when we're reading an abstract, how do we know which one we're reading? Are we reading a full account? Or are we reading something that has been written to try and convince us? And this goes back to the ancient culture that we are working within. That type of person in the 19th century, the independent scholar, was seen by society as being the kind of person that you can trust because they come from a certain stratum of society. And that kind of person is honest, decent, and so on. Now, I'm not sure that was ever true, but that's the story that we tell. And so we rely on trust in individuals. We rely on people being honest and transparent and complete in their account of what they did. But it's not the 19th century, it's the 21st century. We live in a hyper-competitive environment. There are too few jobs. We need to publish and get grants to succeed and to survive. So to what extent is that story we tell ourselves, about being honest and trustworthy and so on, going to survive in the context of that environment? So how can we move from a system that relies on trust in individuals to a system that is more inherently trustworthy and transparent? So I think research culture, as well as those cognitive biases that we all bring to our work as humans and the incentive structures that we work within, is another part of what we want to consider. The behaviors, the values, the expectations, the attitudes. When you talk about research integrity, you can talk about the ethics and governance, the managing risks. You can talk about the professional behaviors of the individuals concerned, and you can talk about the research process itself. And I want to focus on the research process. That is what we as the UK Reproducibility Network, and I think the other reproducibility networks, have as their main focus. We need to understand the other parts, but our role is to improve the research process. And we want to move away from this 19th century model where everyone looked like this. This is not a particularly diverse community. And it's not just non-diverse in terms of all being male and all being white. These were all, nearly all, affluent people drawn from a certain stratum of society, and that created the myth of the trustworthy, honorable, independent scholar who was pursuing knowledge for the sake of pursuing knowledge. In fact, there have always been egos and competitions and so on, but that's the story we tell about ourselves. So we need to move away from that and into a 21st century culture that is more professional, more transparent, and where the processes are more inherently trustworthy. It's worth remembering that many of our ways of working are really ancient. The Journal of the Royal Society, the Philosophical Transactions, is the first. I have to be very careful when I speak to a French audience about this. The first scholarly journal was a French journal. The second scholarly journal, published a few months later, was the Philosophical Transactions of the Royal Society. This is back in the 1600s. But the Philosophical Transactions is the only one still in print. And so you can see a recent version of that journal on the left and the first issue of that journal on the right. So this has been in continuous print for 400 years. And our model of scholarly communication basically hasn't changed over that time. We still disseminate knowledge on dead trees. The way in which we constrain articles is predicated on paper being expensive. It's a very old way of communicating knowledge. So let's focus on the research process. What can we say about the research process and what can we improve about the research process? So a few years ago, a funder from the United States, the CHDI Foundation, which funds Huntington's research, convened a meeting because they were concerned about these issues of research quality. And the discussions that came out of these meetings were published as this article in Nature Biotechnology. The CEO of the CHDI Foundation made the analogy that scientific research, academic research in particular, is like the US automobile industry in the 1970s. So in the 1970s, hundreds of cars would roll off the production lines in Detroit every hour. Productivity was very high, but quality control only happened at the end of the process. Someone would count the number of wheels, check the engine started. And this was an era of very badly built cars that broke down all the time. Because if you got to the end of the process and the car was badly built, there was nothing you could do about it by that point. And often you didn't find out about the fault until the car had driven off and then broke down later. A statistician, W. Edwards Deming, took the concept of quality control throughout the manufacturing process to the Japanese automobile industry, and it was embraced there. They introduced spot checks throughout the manufacturing process. If the error rate was above a certain threshold, the process would stop. They'd identify and correct the source of the error, and then the process would restart. So they moved the quality control from the end of the process to the process itself. Not surprisingly, that improved quality. The Japanese industry still has a reputation for reliability and building reliable cars today. All other automobile manufacturers, in fact, all other manufacturing industries now adopt the same approach. The less intuitive insight that Demings had was that by focusing on quality, not only do you improve quality, but you also improve productivity, because it's easier to stop a process, correct it, and then restart it than to find out that there's an error at the end of the process and then have to go and find it somewhere in the process. So productivity improved as well. Now, if the analogy holds, in scientific research in academia at the moment, the quality control is at the end, it's peer review. There's no quality control throughout the process before you get to that point. And as a result, false findings like the serotonin transporter gene study make it into the literature, and it takes a long time to identify where the flaw was in all of those studies that emerged over time. And as a result, we advance knowledge more slowly than if we were producing higher quality outputs in the first place by ensuring that the research process was high quality. And the approach that Toyota adopted became known as the Toyota model. It had these principles in it. I think these are good principles for academic research culture as much as manufacturing, as much as anything else. And the key one at the center is that the right process will produce the right results. If we can improve the research process and ensure that that is robust and include quality control checks on that process, then we can be confident that what comes out of that process will also be high quality. I think the other points are also worth reflecting on, but I don't have time to talk about all of them, unfortunately. So how does that look? Well, I think the candidate gene literature and the story of the serotonin transporter gene is an illustrative case study for another reason, which is how that field transformed with the introduction of genome-wide association studies, which I mentioned briefly earlier. It wasn't just the serotonin transporter gene that was problematic. That's the story I tell because I was familiar with that literature. Nearly all candidate gene studies produced spurious findings. And we know that because in the early 2000s, the technology emerged that allowed for the simultaneous testing of 500,000 genetic variants across the whole genome in an agnostic way. You didn't have to look in specific genes, you could just test everything at once. But to do that required very large sample sizes, particularly because it was becoming clear that the effect sizes for the association between common genetic variants and behavioral phenotypes, disease phenotypes, were small. So to achieve those very large sample sizes, people needed to collaborate. And in order to collaborate effectively, people had to develop new norms of publishing and agreed frameworks for authorship that moved away from just a list of authors to defined contributions by individuals on that authorship list, which I think was positive. But also, they had to share data and share code. And as a result of that, work in ways that were interoperable and embed quality control checks in their data processing pipelines to ensure that the data from the different contributing studies were all equivalent and comparable. So the technology drove collaboration, and the collaboration drove these new ways of working, which mean that the results generated by genome-wide studies are absolutely robust. This is the first, an association between the FTO gene and body mass index, and this finding is one of the most replicable findings in science. It's absolutely robust. You can now look specifically at this gene in a much smaller sample of a few hundred people, and you will see an association because it's real. And if you don't see an association, that's just by chance, and it hardly ever happens. This is an absolutely robust finding. And the vast majority of findings from genome-wide association studies are robust. Now, what they tell us, what we can do with that, whether it's useful knowledge, that's a different question. But the actual empirical findings are much, much more robust than almost any other domain of biomedical research. And I think we need to think, why is that? What happened in that field to lead to that robustness? And I think it's large sample size, it's statistical stringency, the p-value threshold for declaring discovery is 10 to the minus 8. And it's the incorporation of these quality control checks, the transparency around data and code, and so on. So I think that open research can be a mechanism for quality control. And the reason why the UK Reproducibility Network and others are focusing on open research is that it allows for more scrutiny. People can check your data, check your code, check your study protocols, check your materials. Again, humans make mistakes, humans make typos. There are typos in manuscripts, there's typo in code, there are typos in data sets. We've been publishing in our research group our data sets on the University of Bristol repository for about 10 years. About 3 months ago, a master's student from the University of Glasgow emailed us and said, "I'm really sorry, I found an error in one of your data deposits." And that was scary. But what she had been asked to do for her master's was to find a data set, download it, run the code, and see if what she got was the same as in the published paper. And in our case, it wasn't. One of the tables looked different. So we looked at this, and it turns out she was right. There was an error. Fortunately, the error was just in the data dictionary where one of the variables had flipped. So the data were correct, the code was correct, the published paper was correct, but the data dictionary in the deposit was incorrect. So we were able to correct it and update the deposit. That's good. That's science self-correcting. It was slightly embarrassing, but at least the error was detected. And fortunately, it was a minor error. So that transparency and then incentives for people to check by, for example, embedding these things in undergraduate courses or postgraduate courses, which I think is a really good learning experience, allows for those errors to be detected and corrected. But of course, we need to then accept that there has been an error, not be defensive, admit the error and correct it. So all of the aspects of open research, I think, lend themselves to creating more scope for errors to be detected, and also in turn then creating an incentive for us to ensure the quality of our deposits, for example, because no one wants someone else to find an error. That's embarrassing. We'd rather find the errors ourselves as much as possible. But we have to recognize in any human endeavor, there will be error. It will be honest error the vast majority of the time. And rather than point the finger of blame at individuals, we should think, how did that happen in the process? What went wrong in our process that allowed that error to make it through? And this is an example of what that kind of transparency can do. So in 2000, the National Heart, Lung and Blood Institute, which is the funder in the United States, mandated the pre-registration of primary outcomes on clinicaltrials.gov. So before 2000, if you wanted to, if the results for your primary outcome were not so exciting, you could promote a secondary outcome and in the write-up of your papers claim that that was your primary outcome, and no one would be able to check. So before 2000, each of these dots is an individual study. They're all randomized controlled trials. Before 2000, most of the studies were positive, showed a benefit for the intervention over the comparator. Those are the little pluses. Some of them were neutral, showed no evidence of a difference. After 2000, if you switched your outcomes, someone could go and check, and if you got caught, that would be embarrassing. After 2000, nearly all of the trials were neutral, a couple were positive, and one for the first time showed harm. And the reason I think this is important is that, first of all, it shows that transparency and that potential for error checking can change behavior in a positive way to more honest reporting. But it also shows the problem that early career researchers face, because most research conducted by early career researchers, and I include myself in this when I was a PhD student, looks like the figure on the right. Most of our experiments don't work. But what they see in the published literature looks like the left. Most experiments work. And that creates, or can create, the sense that I'm not cut out for a career in science because my experiments don't work and everyone else's are perfect. And it's not true. The problem is not you, the problem is the literature and how we publish and publication bias and so on. So I think transparency is healthy, not just because it can correct errors, but because it gives people a more honest sense of what research is really like. So how can we then use open research to create quality control frameworks to ensure that those errors are caught early in the process rather than later on? This is where the philanthropic donation that we received is being used in Bristol to create a quality assurance framework within the life sciences that draws on the frameworks that exist within the pharmaceutical industry at the early stage, drug discovery stage of their research pipelines, with foundational training in core data integrity skills, including transparent, reproducible workflows, and a framework for ensuring data integrity and quality assurance based on training, transparency, documentation, standard operating procedures, and so on. And the idea here is that if we can focus our attention on the quality of the process and do that in a way that is consistent across different types of research conducted within the life sciences, we can improve the quality of our work. The hope is also that we can then work more with industry, because when they come in to look at the research that they may want to take forward into their own research programs, they can be more confident in the integrity of the data that we've generated. So this has different elements to it. I've already talked about research culture, so we need to incentivize the uptake of these quality control measures through incentives, promotion criteria, training, awareness, and so on. We need to have the infrastructure in place, the research data management infrastructure, the tools, the policies, the systems. We have a repository at Bristol for depositing data, for example. And we need to have this project, the Reproducibility by Design project, that brings all of that together and uses early adopters of this framework as advocates that can then convince other researchers to change their behavior. Because as we all know, the people who come along to meetings like this one are generally the people who are already convinced that we should be doing things differently. It's the people who aren't in this meeting that we need to be convincing. So how do we do that? And the principles of the Reproducibility by Design part of the project are that research data should conform to the highest possible standards of data integrity, that quality by design and open research principles should be applied from the outset. One of the key things about open research is that it's much harder to do after your study has ended. You should be thinking about it up front, not just in terms of pre-registering your study protocol, but setting up the framework for your data and code to eventually be transparent, consenting participants for data sharing early on, etc. And we need to be identifying which elements of the research process are the high risk elements. We can't look at everything in close detail. So where are the threats to the integrity of our data? It could be our randomization procedure. It could be the assays that we're using if it's a biological study, the reagents that we're using. Where are the threats and how can we ensure quality in those specific areas so that we can mitigate those risks? And so the Reproducibility by Design project works across the different phases of the research lifecycle. At the design stage, you'll see, for example, that the open science plan comes in right at the beginning with a view to eventually making the outputs of the research, including the intermediate outputs like data and code, available. In the conduct, identifying the critical data and the critical processes, like I said, how can you be confident that your randomization procedure is robust and applying proportionate risk management there, but also creating transparent records throughout the research process, for example, through the use of electronic laboratory notebooks, the analysis, coding quality, statistical rigor, independent advice from statisticians, pre-publication checks. There are a few pilots running in the UK on conducting checks on manuscripts before they get submitted to journals. And one of those found a 70% error rate in manuscripts that were submitted for checking. Most of the errors were minor, but again, it speaks to this point that mistakes, typos will creep into our manuscripts, and we need to have a process for catching those early. And then the longer term retention of those open research outputs, the data, the code, and so on. And you can see that this is quite detailed. So at the design stage, which is the most developed part of this project, we have all of these different examples of activity in terms of guidance, training, examples, use of appropriate experimental design, assessing risks to quality, plans to make data and code fair, findable, accessible, interoperable, reusable, and so on. We're still working through the other phases of the research project. We have pilot studies ongoing at the moment to look at the conduct of research, for example, in particular, how we can monitor data quality in an ongoing way, how we can monitor risks and threats to data integrity, and so on. The analysis stage, again, this is still in development, but we know that funders in the UK, for example, mandate data sharing. So we need to have a good data management plan. We need to be applying open research principles. We need to be reporting transparently, and so on. And then at the publication stage, I've already mentioned the pre-publication checks. Those are running at the moment across those institutions shown at the bottom, open access publishing, but also other ways of working that are not directly about the quality of the research, but about the quality of what people get out of the research. For example, recognizing a range of different contributions, creating incentives for people to work in this way through hiring and promotion criteria, and so on. For example, the University of Bristol now includes open research within its promotions framework. If you can explain the ways in which you have worked to make your research transparent, that counts towards your promotion case. And then finally, retention. And this is a challenge because retention is expensive. You need to have a repository. You need to have the high performance computing infrastructure behind that to support it. You need to have teams in the library that check your data deposits before they're published. As I say, we've been publishing our data for about 10 years, but we still sometimes get a data set sent back to us with a query about whether we need to include a particular variable because it could potentially be re-identifying. So we have professionals that check our data sets, that know what they're looking for, that can catch those errors before they get into the public domain. One of my concerns about, for example, the Open Science Framework, it's a fantastic piece of infrastructure, but many of the data deposits on there, because they're uncurated, because no one has necessarily checked them, are not all particularly usable, don't have all of the information that you need in terms of the metadata, data dictionary, and so on. And as a result, are not necessarily that usable, but also might be including information that shouldn't be in the public domain because it could lead to re-identification of participants, for example. So I think those teams are part of the infrastructure as much as the digital infrastructure. So this is all why we set up the UK Reproducibility Networks and why there's been such interest in setting up other reproducibility networks. There is a manifesto for reproducible science that I published in 2017 that identified various threats to the hypothetico-deductive cycle. What we identified were some potential solutions or proposals that could address these threats, and critically the stakeholders that could address these, the funders, the publishers, the learning societies, but also researchers themselves. Open research is one area where researchers have a great deal of agency and can simply choose to start working in a more transparent way and hopefully improve the quality of their research as a result. But really, to bring about change, we need to bring together researchers, institutions, funders, publishers, and create a better system that catches these errors, incentivizes the right behavior. Another great quote from Deming is that a bad system beats a good person every time. And another area that we try to learn from is safety critical industries, which embed what is known as a just culture, where if a mistake is identified, we don't seek to punish the individual, we seek to identify how that system allowed that mistake to happen. So in aviation, in surgery, there are incentives to openly report, call out errors and problems that are identified so that those can be corrected, in part because if you don't do that in those contexts, lives are lost. But we can learn from that and create more of a culture of honesty and transparency, not just in how we work, but in how we talk to each other, in terms of when we call out errors, when we identify mistakes. And one of the things that we've done within our research group is set up a Slack channel called Triumph and Disaster, where people talk about their successes, grants that are funded, papers that are published, but they also talk about their failures. They talk about the grants that were rejected, the experiments that didn't work, again, to normalize the fact that that is just a feature of science, that things don't often work, and that if we don't make that transparent, we can again have that feeling that everything else for everyone else is going brilliantly, but our own research is struggling. So in other words, let's make the denominator of our experience, the number of grants not funded, the number of papers rejected, transparent as much as everything else, to try and foster that climate of being able to talk openly about error. So that is very much what the UK Reproducibility Network is trying to do, bringing together all of those different players in the research ecosystem, the researchers themselves, the technicians, the librarians, the institutions, the incentive structures, the funders, the funder mandates, publishing policies of journals, and so on. And as Davide mentioned earlier, this is growing across the world. And in fact, tomorrow I'm off to Ukraine, which is quite exciting, for the launch of their reproducibility network in Lviv. We have a regional network in Africa because the individual countries within Africa typically don't have the critical mass to support their own network. So there we have a regional network. And you can see that we've grown rapidly across Europe. And in fact, this is out of date because we have a Danish reproducibility network as well. It's not shown on that figure. And that to me is very exciting, but it also illustrates that there is clearly an appetite for this kind of collaborative approach that is different to the hyper-competitive nature of academia that we normally find around us. And I think we can achieve a great deal working together collaboratively in this way. Thank you.
Thank you very much, Marcus, for this wonderful presentation. It was really insightful. And if there are any questions from the audience, here we can start from with our Q&A session. And so if you have a question, you can speak up, or if you prefer, you can type down your answer into the chat, and I will read it to the speaker, to Marcus.
If I may, sorry, my webcam is not working. Buongiorno, Marcus. I have just a curiosity. I'm a big fan of ethics and deontology, and I was wondering whether, for example, in UK, you have any teaching of ethics and deontology for PhD students before? I mean, because my idea is that, for example, we start doing research without being told how we should do research, and especially the limits we should have. I mean, all the things you said in your talk, basically. I mean, we have never been trained or have never been trained in ethics and deontology before starting doing science. Okay.
Yeah, I really agree. And we are trying to introduce some of that. We have courses on research integrity that include elements of that, but I think we could do a much better job. Many people, and I include my senior colleagues in this, don't really understand why we have ethics committees. What happened in the 1940s and 1950s that led to the realization that actually we shouldn't be policing ourselves. We should have someone independent look at our research and check that it was ethical before we can approve it. And we don't really teach that, in my experience, and we don't teach it well. And we don't teach this culture of ensuring that the research that we produce is worth producing. It's not just about the quality of the research. It's also about whether or not we should be spending time, effort, public money on a research question in the first place. I edit a journal, Nicotine Tobacco Research, where we now get, usually from developing countries that are trying to grow their research system, studies that say smoking is associated with lung cancer. And it's a well-conducted study in a large sample with adjustment for confounding and so on, but we know the answer to that question. We don't need another study. All it does is create a burden on the system, more reviewer time, more editor time, more journal pages, and so on, without advancing knowledge. So we seem to have lost sight of the fact that fundamentally, what we do should advance knowledge. And that can be basic knowledge, it can be discovery research, it can be applied knowledge. But if we're simply, and obviously I recognize the importance of replication, but at some point you get to the point where you don't need another replication study, you know the answer to the question, it's absolutely robust, and we should be doing something else. So I think there is a lot more that we could do with early career researchers, with PhD students, around ethics and around these kind of cultural issues more generally, to try and instill those values early on, and hopefully then retain those values throughout the system.
Thank you, Marcos. Davide Serpico had a question also.
Yes, thanks. Can you hear me?
Yes, we can.
Hi, Marcus. Thank you so much. It was brilliant. I have a question about the relationship between replicability issues and sample sizes in the context of genomics and quantitative genetics. Although I agree in general with your analysis, I was wondering whether there is any other source of low replicability, publication biases apart, concerning, for instance, heterogeneity in population stratification issues. So you mentioned the two cases. One is the FTO gene for variation in body mass index, and then the genes for serotonin transporter in gene environment interaction studies. So my worry is, we know that for many complex traits, there is much variability about how clinical symptoms and phenotypes are like realized in different individuals. The same symptoms can have different genetic bases, and the expression of genes can depend on environmental factors. So I was wondering, perhaps having larger sample size, some argue, can introduce more uncertainty, since there is more stratification, more variability within the same sample, and we are analyzing more individuals with the same phenotype, but we are not sure exactly whether they are etiologically similar enough to make findings consistent. So I was wondering whether there is any way to save, in some sense, the results of previous studies, like those on the interaction between some genes and variation in depression, because perhaps those studies didn't identify widely generalizable results, but those results were relevant perhaps in some subpopulation that contingently was picked up by the small sample size. I don't know, perhaps there was something statistically relevant in those studies, but not as widely generalizable as we would expect. What do you think?
Well, there's a lot in there. I mean, first of all, genetic studies historically have focused on samples of European ancestry because of population stratification, which is that the prevalence of different genetic variants is different in different parts of the world because of how populations have moved around over long periods of time. And that can create confounds if there are also differences in behaviors across different parts of the world.
Sure.
So to deal with that, initially, samples were restricted to populations of European ancestry. We now can adjust for population structure in those analyses, so the technology has advanced. So I agree that that's an issue more of generalizability than anything else. To what extent are the findings in a sample of European ancestry going to generalize to a different population? In terms of whether or not some of these findings could be rescued, I think I would be skeptical because in the context of gene-environment interactions, which certainly exist, I'm not denying the existence of those, almost always, with sufficiently large sample size, that interaction would be reflected in a main effect. This was the focus of our 2008 paper. Only if you have a perfect crossover will you not see a main effect. And most of the gene-environment interaction results that are presented are not perfect crossovers. So you would eventually find a main effect. And if you're not finding a main effect, then that suggests that the interaction that you're seeing is probably spurious. So I would say the best approach is to use genome-wide methods to identify your candidates where there are main effects, and then look for interactions between environmental exposures and those genetic variants, and do that in a way that takes into account population stratification and so on. So I think you're right in the points that you make, but the approach that I would take would still be to rely on the kind of empirical approach with large sample sizes to identify your candidates and then take those forward. And the problem was that with candidate gene studies, we did it the other way around. We looked in areas where we thought there might be something happening. Serotonin, we know, is relevant to anxiety and depression. Therefore, we look at the serotonin transporter gene. And actually, one of the interesting things that has come out of genome-wide association studies is that very few of those candidates came up in those genome-wide association studies, which I think tells us how little we know about the biology of behavior. That the areas that we were looking were just not the right behaviors. And in some senses, that's exciting because it means that new avenues for exploration have been generated by these approaches.
Thank you. Yeah, it makes sense. Yeah.
Okay, thank you again. Are there any other questions? So,
and what I would say is, please do email me or contact me on social media, however you want to. If you have any follow-up questions, I'm more than happy to answer more questions, share papers, share my slides, if that's useful, so on. But thank you again for the invitation.
yeah, that would be very, very useful. As you know, most of the audience are PhD students. So I think that you provide some very insightful tips on how to start doing research and start doing it as a profession. So thank you very much. And let me please, if there aren't any other questions, please I'll
say one more thing very quickly.
Yeah.
So if a lot of people here are early career researchers, I completely sympathize with the need to make sure that you have a career. However much people go into science because they're passionate about the subject, they also want a job at the end of the day. What I would say is that increasingly, certainly in the UK, I don't know the Italian system so well, working in these ways makes you more employable. You can put more evidence of completing projects on your CV, data sets, code, pre-registrations, preprints. They're all completed tasks that evidence your ability to complete something and allow you to demonstrate your personal contribution in a more granular way. There may be a 3-experiment paper and you generated the data for experiment 2, and you can make that a deposit and therefore publication and list it on your CV. So it allows you to demonstrate your skills and your ability to complete tasks better. And increasingly, because more and more funders are asking for these things, PIs are looking to recruit postdocs who can do these things, who have these skills, and who can teach their labs how to do it. It also makes you more attractive if you want to pursue a career in industry, for example, because again, you have these relevant skills. So there's a moral argument for working in these ways, I would say, But there are also pragmatic arguments around improving the quality of our research and the interoperability between research groups. And there are also selfish arguments that this is better for you and your research and your employability. So I think it's worth bearing that in mind as well.
So, thank you. Thank you again. Let me share my screen. So I would say a few final concluding words. Can you, okay. You may have seen on your screen a Windows popping up. So if you need a certificate of attendance for this seminar, you had just to type your email into the poll, and then we track all the answer and send you the email, certificate email within the next 7 days. And let me introduce you to the next seminar, which will be on the 11th of December in the afternoon at 3 p.m. And we will have Tony Rosselhauer, who is going to talk us about large language models, open science, and trust in scholarly work. And finally, if you want, you can see this seminar and last seminar, previous last year's seminars on open science promoted by ITRN. So if you want to know more about open science, reproducibility, we touch a very wide range of topics in the last year. So if you are curious, you can check on the YouTube channel and also subscribe where you land there. And finally, we also landed on other socials. So we have an Instagram account, a Facebook account, and a LinkedIn account. So if you want to get in touch with us, with our content, and also our new initiatives, please stay in touch with us using the social. So thank you very much again for having you here. Also, thank you, Marcus, for the seminar, for today's seminar, and have a good day.
Bye-bye.